Okay, here we go. Welcome to Biology 1011. This is Principles of Biology Part 2. My name is Jillian Guess. I'm the course coordinator for the class. So I'm the person to ask if you've got any administrative type of questions, if you get sick and are going to miss the midterm, something like that. Todd Bishop, who lots of you know from last term, is also part of the class, just not here today, and you'll see him in the lab. So he's a good person to ask, especially if you have questions related to the lab. Dr. Mark Johnston, who's sitting over there, is going to be giving the lectures in our first unit of the class on plants. So my goal today is to tell you a few quick things in a few short minutes so that we can get onto plants. Okay. As we set out, let's start with an acknowledgement. Dalhousie University Senate acknowledges that we are in Mi'kma'ki, the ancestral and unceded territory of the Mi'kmaq people, and pays respect to the indigenous knowledges held by the Mi'kmaq people and to the wisdom of their elders, past and present. The Mi'kmaq people signed peace and friendship treaties with the Crown, and Section 35 of the Constitution Act, 1982, recognizes and affirms Aboriginal and treaty rights. We are all treaty people. We recognize that African Nova Scotians are a distinct people whose histories, legacies, and contributions have enriched that part of Mi'kma'ki known as Nova Scotia for over 400 years. Here's a photograph of a hemlock forest in that part of Mi'kma'ki known as Unama'ki, or Cape Breton. So before we set off down that path toward plants, I have just a few bits of information for you, just to help you get started in the class. The course syllabus is published, it's posted on Brightspace, and I would like all of you to take the time to read it carefully. I'm not going to talk about everything in there today, just a few highlights, but it's got a lot of important information that you need to know. If you are taking Biology 1011 this term, but you did not take Biology 1010 last term, first of all, welcome aboard. Second of all, please on Brightspace find the document that's called for students starting in Biology 1011. That's in the syllabus or textbook orientation folder, and that's going to give you some information that you need just to get you kind of a little more oriented to the class since you were not in the first half of the class last term. And you can contact me or Todd if you have any questions after you review that document. Okay, a couple quick topics. First of all, about access. The Student Accessibility Center, and there's a link there so you can find your way to their site, is our center of expertise for anything related to student accessibility accommodations. So if you're encountering barriers in any aspect of your classes, get in touch with them. If you have approved accommodations, please don't hesitate to make use of them. And if you've had adaptations or accommodations, say in a previous university or in high school, and you're not sure about getting those in place at DAL, it's worth making an appointment with an access advisor as soon as possible, because the process takes some time. So get started even if you don't yet have the documentation you need, or even if you're not sure what that documentation is going to be. Just get started. On the subject of access, I will say we do record the lectures in this class, and we work to have an edited and captioned version of that recording available within 1 week of the scheduled lecture. So for those of you who were with us last term, you'll remember that we did that, and we're lucky to have the same student helping edit and caption the lectures, which were really well done last term. So we're hoping to provide that as well. A little bit about exams and communications. Biology 1011 has 3 units. And I'm here to talk about the lecture section. Of course, there are labs associated with each of these units too, to give you the whole class. Unit 1 is plants, lectured by Dr. Mark Johnston. Unit 2 is ecology by Dr. Suchita Arif. And Unit 3, Isabel Abe will teach you about animals. Your midterm, you have a test coming up early next month. So make a note, this is in the syllabus as well, of course. Wednesday, February the 7th, in the evening, is going to be your Biology 1011 midterm. I'll have more detail about time and location as we get closer. But that's going to cover Unit 1 only, plants lectures. Then there's a final exam for a further 40% of your course grade, and that covers Units 2 and 3 only. That's going to be scheduled during the formal exam period. It'll be scheduled by the registrar's office, and we'll let you know as soon as we find out when it is. But in the meantime, please don't make any travel plans during that designated exam period until you know for sure what your exam schedule is going to be. And then the final 40% of your grade comes from the labs. As far as communication, Please do make sure to check your DAL email regularly and check our Brightspace site regularly. The announcement section, which is like front and center, that's where we're going to convey important information to you about the class. So we do rely on you to check that regularly. It's winter term, so we have this special consideration that we get a lot of storms around here. For example, tomorrow we may have a storm. So if you're wondering whether DAL is open or not, I'm not the person to ask for that. Please, in that case, check the DAL website, and actually you can get yourself signed up for text messages that will inform you when campus is closed and open. So in winter term, we always have to keep our eye on the weather a little bit and deal with that form of uncertainty. Labs and quizzes. There are no labs this week. Labs begin next week, the week of January the 15th. Our lab rooms are the same rooms we had last term, LSC 2097, 2098, and 2100, but your particular lab room might be different than it was last term. So please just check your schedule carefully and make sure that when you show up next week, you already know which room to go into. You'll need to have a lab manual, of course. Still quite affordable, $9.42, available at the bookstore just across the way. So please be sure before you come to lab next week that you have a lab manual and you want to bring that with you to every lab. There's an orientation quiz that is a test of your knowledge of our lab policies and some safety information. That's worth 2 points on your final grade, which is not nothing. So you're going to want to make sure to complete that, and that's available now. As well, each of our 8 labs has a pre-lab quiz associated with it. And each of those quizzes are going to become available on Brightspace 8 a.m. Friday, the Friday before that lab week, and it will remain open until Monday, 8 a.m. of that lab week, regardless of what day your particular lab is on. Did you have a question? Good question. The question was, if you completed the orientation quiz last semester, do you have to redo it this semester? And the answer is yes, you do. I bet some of the questions will feel quite familiar. Yeah. So yes, even if you completed the lab quiz last term, the safety orientation quiz, make sure to do it again this term. So as to pre-lab quizzes, your lab, since lab 1 is next week, this coming Friday, 8 a.m., that's when your lab 1 pre-lab quiz will become available. To get ready for those quizzes, for the orientation quiz, you're going to need to read the safety rules and the lab policies section of the lab manual. And for the Lab 1 quiz, you're going to need to read the Lab 1 section of the lab manual. What we've done, just in case there are people who don't yet have a lab manual, is we've posted both of those sections as PDFs in the labs folder on Brightspace. So if you don't have a lab manual yet, first of all, make sure to get one. But second of all, you're still in a position where you can prepare for and complete these quizzes. So please make sure that you do that. As far as that information goes, are there any questions? Okay. And all of our email addresses are in the syllabus, right? So if you have a question, email is a good way, or you can find us in the lab area when labs are in session and ask that way. Okay. In Biology 1010, which is the course last term, the last unit that we did, the last section of the class, was about evolution. And Charles Darwin, in his book on the origin of species, this book ends with this very famous, beautiful, striking paragraph where he asks the reader to sort of use their imagination and envision what he calls an entangled bank. So if you just picture like a steep hillside or maybe the bank of a river or something, and he says, picture this entangled bank clothed with many plants of many kinds, and living in and around the plants and within the soil, this great diversity as well of animals. And he says, think too about the interactions between these different living things and between these living things and the environment in which they live. He says they're dependent on each other in so complex a manner. So that was kind of our ending point in Biology 1010. And now in the class this term, you're going to have the chance to learn a lot about this incredible diversity of animals and plants, their structures, how they work, their physiology, how they make their way in the world, and the kinds of interactions that they have with one another, with the environment around them. Today, in Biology Today, we study living things with this sophisticated set of tools, from molecular tools to microscopes to mathematics and computation. And we do this in order to build up our understanding of life, of living things at this wide range of levels of organization, from smallest to largest scales. So it's going to be really wonderful. And I think we should get started. Dr. Johnston is your guide through plants. He's also the chair of the Department of Biology and a past winner of the Faculty of Science Award for Excellence in Teaching, so you're in great hands. Thanks, everybody.
Good afternoon. I'm Mark Johnston. This is the plant section of the course. It's the first third of the course. I'm actually going to tell you about plants and fungi. I'm going to give you one lecture on fungi. Fungi are more closely related to you than they are to any plant. So why in the plant section? Well, a long time ago, many, many decades ago, fungi were studied along with plants often, probably because they sit still like plants do. But they're not at all related to plants. But they're really cool. And so I'm going to introduce you to them. No one else will, so I'm going to do it. So yeah, it's a course about plants. We're going to talk about a number of different topics. And I'd like to describe how I think you could do really well in my section of the course. First of all, take notes. Taking notes is shown by study after study after study, without exception, to engage your brain in a particular way that makes you learn something. So if you've not taken notes before, or you think you're not very good at it, I would say give it a try. Try really hard to take notes. There's something about taking notes that makes you remember things and sometimes even understand them at the time. It also gives you the opportunity to then to look at your material again. It turns out that writing things with your hand is better than typing, but both work. So first of all, take notes. Second, to the degree you can, turn off distractions. There will be someone in front of you looking at something that is distracting, right? Something having nothing to do with this course, right? Try not to look at that person's screen, and don't do it yourself if you can avoid it. This is a problem for everybody. It's not a problem for students only. It's not just a particular generation. It's a problem for the whole world right now. So to the degree you can turn off your distractions, do that. Don't keep thinking about what's on my phone, what's on this, what's on that every few seconds. That's a guaranteed way not to benefit from being in a classroom. Use the resources. So I'm going to give you a number of resources. As you know, there are slides for this that I show you in lecture that I put up beforehand, at least the day before. I also give you something I call a study aid. The study aid contains some really important information. It contains a complete outline of my lecture. So unlike this brief outline that I show you in class, it's a very complete outline. It also contains a set of questions that you can find in your textbook. There are answers there as well in the textbook, so if you want to look at those, you can. And most importantly, perhaps, it contains every scientific word that I think is important. So I list every scientific word that you should know for my section. I'm also going to put up later on, toward the end of my lecturing, a set of practice questions. It'll be a total of more than 70 practice questions that you can use to practice for the midterm. So you can get a sense of exactly what sorts of things I'm going to ask you, how difficult the questions are, the style of the questions, the material covered, etc. As you just heard from Jill, the lectures are recorded. So a week after lecture, more or less, someone will be able to we'll put those up and you'll be able to have a look at those. I also want to emphasize that in my section of the course, I will only ask you material that I've talked about in lecture. I will only ask you material I've talked about in lecture. If I don't talk about it, I'm not going to ask you it. So you don't need to go hunting around in a textbook for something and worry about something I didn't mention. The textbook. It's there, it's good. There are lots of good biology textbooks now. Of course, it's a huge market. And so, avail yourself of the textbook readings. I have a list of readings that I've posted, so read before or read after if you like. But that material is there for you to have a look at as a supplement to what I'm telling you about. So it's a different way to be introduced to that material. So today we're going to talk about plants in a very general sense, and then we're going to ask what is a plant, what are the ancestors of plants, and then we're going to get into this group that we're going to spend the rest of the plant thing on until we get to fungi, and that's the land plants. It's a subset of all the plants, and it's the dominant set of organisms, the land plants, the things that live on land. All right, so how do you do well in this course? If you were taking notes, you might have written down something like this. I'd also like to mention that Jen Van Dommelen has organized a set of drop-in sessions, and I think she did this last term. And so the first one is on the 12th of January, and there's no registration needed. You just drop in, and it's in the LSC 334 classroom. And everybody's welcome, so just come on in, ask anything you want, or sit there and absorb knowledge. So I didn't know that Jill was going to mention Charles Darwin or give a quotation from him, but here's a quotation that wasn't intended for publication. It was just something he wrote in his diary when he was traveling in the oceans off of South America. And he wrote, "I'm strongly induced to believe that as in music, the person who understands every note will more thoroughly enjoy the whole. So he who examines each part of a fine view may also thoroughly comprehend the full and combined effect. Hence, a traveler should be a botanist, for in all views, plants form the chief embellishment." And that's true. No matter where you are on the terrestrial part of the earth, plants form the chief view. They form the chief embellishment. And if you've had experience in this, if you know your plants, you can usually tell what part of the world you're in if you see the plants, either the landscape as a whole or the individual plants. Okay, so plants are so fundamentally important to human life and to the life of all animals, in fact, and of many other sorts of organisms, that we're going to talk about some of those, how they're fundamentally important. And I want to introduce the idea by asking you what you think were the greatest inventions of humans in human history. Sliced bread was one answer, yes. What's another invention in human history? Okay, soap, yeah, okay, hygiene, yes. Telephone, okay. Electricity, keep going backward. Back in time. The wheel, yes, back there. The wheel, okay, yeah. What's that? Agriculture, yes, thank you. Agriculture, absolutely. What about cooking and fire? Controlled use of fire. What about language? That's about as fundamental as it gets, right? What about the internet? You have no idea what life was like without the internet. There was a universe without the internet. You know, there was one. It actually existed. That was fundamentally changed the world. Agriculture, yes, that's right. So, agriculture is the growing of plants and animals in one place. That's what it is. It was invented on multiple occasions in various parts of the world. Wheat, for example, was first grown in an agricultural setting in the mountains of southern Turkey about 10,000 years ago. Rice was invented as an agricultural product in Southeast Asia, in India, and in Africa, separate occasions. Different species in Africa and Asia. So, why is agriculture important? It fundamentally changed human history. Agriculture allowed humans to settle into permanent villages, which then could become bigger towns, cities. Agriculture, therefore, allowed humans to have a larger family. If a family is not traveling around following the seasons for food, following where the animals are for food, then you can have more offspring. If you're traveling around, how many offspring could an individual take care of who's highly dependent? One, right? Well, if you're settled into one place, you can have multiple offspring that are highly dependent at one time. This led to a diverse economy, less cooperation among humans, among individuals, which gave rise to socioeconomic classes. And, of course, therefore, this idea that here I've got my village, here I've got my field, Here I've got my stuff that I need to eat, my family is going to use to eat, my village is going to use to eat. Here it is, and out there is all the stuff that's trying to harm me. So this idea that nature is somehow separate from us is a fundamental part of humans now, right? It didn't used to be. Thousands of years ago, it wasn't like that. But now we feel that, we got to keep it away and keep all the danger away. So separation of humans from the rest of nature, and of course, other features like large-scale war. So the far-reaching consequences of agriculture, that is the domestication of plants primarily, and also animals. Here's a plant. This is the drink of the gods, the food of the gods. Theobroma cacao is chocolate. This is a tree that's native to South America. And theo means of the gods, and broma means food. So the scientific name for this plant is food of the gods. And what you see here are seeds. These seeds are what we make chocolate from. Seeds you're going to see in this course when we talk about reproduction in flowering plants. Seeds come from fruits, and fruits come from flowers. So here on the left you can see the flowers of the chocolate tree. The flowers of the chocolate tree are weird. They come right out of the woody part of the plant. That's kind of unusual. That's pretty unusual. And then on the right you can see these fruits. You can also see some flowers. But these fruits, each one of these fruits came from a single flower. And each one of these fruits contains seeds that then we turn into chocolate. How did you get here today? You may have driven, you may have taken a bus. If you did that, then the energy that propelled that vehicle was from a plant that lived 300 million years ago. The gasoline, the oil, is from a plant that lived plants that lived 300 million years ago. The tires on that vehicle were made from rubber. That rubber is made from the rubber tree, also native to South America. The scientific name is Hevea brasiliensis, and rubber is made from this milky latex that comes out from under the bark. So you go up to a rubber tree, you slice it open, you put a pail there, and you've got the latex, and then you can turn it into rubber. About half the rubber that we have now, that we produce now, is made synthetically, and about half is made naturally. It's from the rubber tree. The rubber tree native to South America is now planted, of course, across the world in plantations. And synthetic rubber differs from real rubber, natural rubber, in various properties. So tires, for example, have a lot of real rubber in them. Rubber's called rubber. Does anyone know why rubber's called rubber? Joseph Priestley named it. It's because it rubs out pencil marks. Okay, so you may have walked here. If you walked here, you may have been kept warm by clothing, and that clothing could very well have come from a plant. Linen, for example, is from a plant. Cotton is from a plant. Here's cotton. This is a cotton fruit, and the fruit has in it this enormous amount of fiber. And we harvest that fiber and make cotton products, and that fiber is actually primarily cellulose, which is a polymer. Plants are used for drugs. Almost every drug before the 20th century was from a plant, essentially. And now we still discover drugs in plants, and we have often then found ways to make them industrially. So here's a plant that a drug was discovered in, and this is cinchona, or the cinchona tree. And this is an example of an organism that was known by the natives in South America, the high Andes, to be a treatment, a very effective treatment for malaria. There is a form of malaria in South America. And when the Europeans came over, the Europeans learned this, and then they took the bark back, and they took the tree back, and they started to plant it around the world, India, etc., and used this to treat malaria in other parts of the world. They also brought, as you know, people against their will from the old world to the new world, and in so doing, they brought a more virulent form of malaria to South America. That's a different story. So this thing in here that's a treatment for malaria is called quinine. And quinine is what makes tonic water bitter. It's what gives it that wonderful sort of, almost flavor, but bitterness that you use to mix in with other things or drink by itself. And it's used, of course, to treat malaria, but it's also used to treat anemia. It treats muscle spasms. Some people who have cramps will drink it before they go to bed. So it's obviously used in tonic water. And it is an example, quinine, of a plant secondary compound. All the drugs that you can think of from plants are secondary compounds. A secondary compound is a compound that's not used in metabolism and not used in photosynthesis. That's the definition. All the drugs are secondary compounds. All the drugs from plants are secondary compounds. Why were they made? Why would a plant make a compound that's not used in metabolism and not used in photosynthesis? Wouldn't that be a waste of energy? What do you propose? Why would a plant make a compound that's not used in metabolism or photosynthesis? Protection? Yep. Incentive to spread. So to spread what? Okay. Okay, yes. So, yes, something like a scent to bring something to, the idea is to spread progeny. Okay, yeah. And protection. So plants are hanging out in the world and they're attacked by all kinds of things, mostly insects. And every single species essentially has evolved a suite of secondary compounds that it uses to defend itself against a suite of insect species. Here's a drug. This is probably the most commonly used drug by humans. This is caffeine. Caffeine comes from the coffee tree. Coffee trees come from the same place that humans come from, Ethiopia. Here are the coffee flowers. Each flower turns into a seed. Each seed is what you call a coffee bean. You can roast them. Here's a necklace somebody made of the very simple compound called caffeine. And caffeine is a stimulant in humans, and for the coffee tree, it's something it uses to protect against herbivores. Here's aspirin. Aspirin is from the willow tree, or the willow shrub. Willows are a genus called Salix, the genus Salix. And they are quite prominent in Arctic areas. We have them here, of course. But as you go into the Arctic and trees disappear and all the woody plants are shrubby, many of those shrubs are willows. And those willows have acetyl salicylic acid in their bark. So if you're ever walking around the Arctic and you have a toothache, find a willow tree and chew on the bark. There's aspirin in there. If you look at an aspirin bottle, it says acetyl salicylic acid on it. That's what this is. It's in willows, the exact same thing. Real, natural aspirin. And so it's used, as you know, of course, for pain, for fever, for inflammation. Here's another commonly used drug, THC, tetrahydrocannabinol. And this is found, of course, in hashish and in marijuana. And one of the species of marijuana is called cannabis sativa. And here's the plant. And you know that cannabis sativa is one of those 5% of flowering plants that have separate male and female individuals. That's really unusual in plants, and we're going to talk a lot about that. Here's tobacco. This is the source of nicotine. Nicotine is an alkaloid, a type of compound, secondary compound in this case. And here's the plant on the right. You can see the flowers, tubular flowers. They're pollinated by moths. And the dried leaves on the left, getting ready to be processed. Here's the opium poppy. It's the source of a number of opioid drugs. Heroin, opium, for example. And then more in a medical context usually, codeine and morphine. These are very strong, they have a very strong effect on your physiology and your mental state. And you know, codeine is what's added to Tylenol to make Tylenol 3, which is a narcotic. This is Taxol. Taxol is the brand name for a drug called paclitaxel. And this is the most commonly prescribed, the most commonly used chemotherapy drug. So it's from an evergreen plant called the yew, Y-E-W. And you all know what a yew is. A yew is the plant that you see along people's houses, so sort of a hedge around the house or even a hedge around the property. Those are almost always yews, unless they're tall and columnar, then that's something else. But these yews have this little red, often have this little red thing here that you might think is a berry. Technically it's not. It's technically a cone, believe it or not. But this yew is the source. There was a particular species that is the source of Taxol. And it's called Taxol because the genus of yew, the genus of yews is Taxus. This is a rainforest, and this rainforest shows you all four groups of plants that we're going to talk about. So after I introduce plants today, next time we're going to move into talking about four different groups, and they're going to be the mosses, the ferns, the conifers, and the flowering plants. You can see a birch back here, that's a flowering plant. And these land plants make oxygen. So one of the major reasons that plants are important is that they provide oxygen. Half of the oxygen, so every other breath you take, the oxygen in it is from a land plant. And every other breath is from a marine organism. It's all from photosynthesis. All the oxygen you get, the 21% in the atmosphere, is produced by photosynthetic organisms. So this is just a recap of stuff I've said, so there it is in writing for you. Now, if you are a person studying plant biology, you could actually study a number of different topics. So, you know, some of us naturally gravitate toward more statistical things, some of us gravitate toward morphological topics, some of us gravitate toward cell biology, genetics, molecular biology, you know, and sort of the others are less interesting or our brains don't work as well in them. But the study of plants is a study of a group of organisms. So if you study plants, you really are probably studying a subject matter and studying plants for them, rather than studying plants themselves. So here is a whole set of subjects that you could be studying if you called yourself a plant biologist. So I tend to fall in this evolution and ecology area, and so you could study evolution over short time periods, over long time periods, natural selection. You could even study how the DNA sequences evolve, etc. You can study ecology. You can study how organisms interact with one another, how they affect one another, why this organism lives in the desert, why this organism lives in the rainforest. You might study molecular genetics, turning genes on and off. You might study you might do mathematical modeling. That's actually useful in a lot of these areas, evolution, ecology. You could mathematically model leaf shape and how air flows over leaves of different shapes. You could study morphology, which is the external form of an organism, or anatomy, the internal form. Physiology, cell biology. Systematics, the relationships of groups. And development. And we're going to touch on most of these things in our jaunt through the world of plants. Here's an example of the sort of question that one might ask. Here's one of my favorite plants. This is something I've done a lot of work on. This is called the cardinal flower, the ruby-throated hummingbird. And you can see here a hummingbird. This is in Algonquin National Park, or Provincial Park. It's visiting this plant. And it's going around and it's getting nectar, sugar water, from the flowers. And this hummingbird is also picking up and depositing pollen. The pollen contains sperm, and the plant is paying to have its gametes moved. That's what flowering plants do. They pay somebody to have their gametes moved around. So it's paying this pollinator by making very expensive stuff, which is nectar, sugar water. And the pollinator comes, visits, and in this case, the pollinator doesn't care about pollen at all. It's not using it. It's not like a bee. It doesn't care. It's not using the pollen. But it's inadvertently getting it on its head and moving it from one flower to the next or one plant to the next. So it turns out that there's a really cool feature of this plant, cardinal flower, and that is that the flowers go through a male phase first, where they give off pollen, and a female phase second, where they receive pollen and make seeds. So the ones at the bottom are older, so they tend to be female at any given time, and then they'll become fruits. And the ones at the top are male before they become female. So they're younger at any given time. So these are a bunch of male flowers, male phase flowers, and these are a bunch of female phase flowers that used to be male but are now female. So, as you know from royal families, mating with yourself is not a good idea. Inbreeding in general is not a good idea, right? It's not a good idea across the biological world. So, if a pollinator, a hummingbird, were to come along and visit the male phase flowers, get pollen on its head, actually you can see the pollen up there, see the yellow? That's pollen. Get pollen on its head, and then come down and visit the female flowers and deposit the pollen on these, that would be self-pollination. So those offspring that came from that would be from selfing. And selfing is the closest inbreeding possible. It's mating with yourself. It's not mating with a cousin or brother or sister. It's mating with yourself. You know, there's no one closer to you. So that's a type of inbreeding called self-fertilization. So what does the plant want? The plant wants the hummingbird to come in at the bottom, bring pollen from someone else, so someone else can be the dad to these seeds, move up, getting nectar from these things, picking up pollen from the male phase flowers, and then leave. So then you are fathering someone else's seeds, right? That's what the plant wants, in a sense. Come in at the bottom, move up, leave. Right? So let's see what this one does. Okay, here we go. Here comes a hummingbird. Nope. It's visiting a male phase flower. Male phase. Look at that, drinking. You can see the yellow on its head. Drinking. Male. That's a female. So it just took pollen and transferred it within itself. So this is causing self-pollination. I've actually studied the effects of this, and it turns out that self-pollinated seeds are far worse off in growth than outcross seeds are. So they don't do nearly as well. So the conclusion there is it's really tough to be a plant. It's really tough, you know. You gotta face these gorgeous little pollinators that come along and just, you know, do what you don't want them to do. The other reason it's tough to be a plant is you don't move. So, you know, if a lizard doesn't like its environment, it just moves to a different place. You know, it moves to the warm rock, or it leaves the sleet and goes, you know, underneath something. Plants don't do that. If it starts to sleet, you're sitting there and you have to deal with it. You've got to do something physiologically that's different, right? You've got to deal with it. Winter comes, you got to deal with it. You're not moving, you're sitting there. An elk steps on you, you got to deal with it. You know, a caterpillar eats part of you, you got to deal with it. So that's something we're going to see throughout this section of the course, and that is that plants have particular ways of dealing with the environment. In fact, I think this quotation from Shakespeare is really apt. Plants sit still and suffer the slings and arrows of outrageous fortune. So, well, he didn't say plants sit still, but he got the slings and arrows part, outrageous fortune part. So what's this from? Yeah, it's from Hamlet, that's right. Don't have anyone who played Hamlet here? Okay. Okay, so what's a plant? A plant is what we want to call it. We can define it however we want. The important thing is the relationships of different groups, right? So if you want to call a set of things lions or a set of things mammals, you just do it whatever convenient, right? That's fine. The truth, though, is the relationships among the different groups. So once you've settled on a name for something, stick to it. And that will be then your language. So there are a number of different groups that could be considered plants, candidate for the word plant. And everything up here could be called a plant. And the reason everything up here could be called a plant is because they all have something called a plastid. A plastid is an organelle, and that organelle is most famously represented by the chloroplast. And the chloroplast is what carries out photosynthesis. So the plastid originated 1,500 million years ago, that is to say 1.5 billion years ago, 1.5 times 10 to the 9 years ago, here on this evolutionary tree, and it gave rise to these different groups. Everything after this has a plastid. So the red algae, so-called, this set of green algae called the chlorophytes, and this set of things called the streptophytes. Weird words. So the plastid was the engulfing, that is the endosymbiosis, of a bacterium that photosynthesized by another eukaryote, giving rise to everything. So you could call everything up here a plant if you wanted, just for convenience. Okay, so what? It's just a definition. What matters is this organization, these relationships. So at some point, the red algae split off from the rest of these things, which we call the green algae and the other plants. And it's these things here that we are going to call the plants. And a technical term for them is the viridiplantae. And you can see it written there from your textbook. There it is, viridiplantae. So this shading here. So everything I've circled here is the viridiplantae. And for our purposes, these are the plants. So the plants contain these three groups. First of all, the chlorophytes, which is a type of green alga. Second of all, the charophytes, which is a type of green alga. And third of all, the embryophytes, or the land plants. We are going to spend everything after today talking about the land plants. We're not going to talk about the charophytes or chlorophytes any further after today. The land plants are what dominate the planet, dominate the terrestrial part of the planet, and we are going to focus on 4 groups of them. Okay, so I mentioned endosymbiosis and the origin of plastids. It turns out that this origin of plastids that I showed you is called a primary endosymbiotic event. It means that, if you see here on the lower left, there's a eukaryote shown in red if you see red, and in green, a prokaryote if you see green, so there it is. So the prokaryote is a cyanobacterium. Here's the word. Where is it? Cyanobacterium that was engulfed by a eukaryote, and that eukaryote already had present in it a mitochondrion. So this formed a new organism, a special organism, that now had both a mitochondrion as an organelle in it and a photosynthetic plastid called a chloroplast, which became a chloroplast. Fine. So now you've got your red algae and all those other things, right? Great. It turns out that there are a whole bunch of weird organisms that you've probably never heard of unless you talk to Alastair Simpson, and maybe he even presented some of this stuff. There's a whole lot of other weird organisms, mostly single-celled, that took this thing, this new plastid-bearing eukaryote, and engulfed that. So then, this is called secondary symbiosis, and this is where a eukaryote engulfed a plastid-containing eukaryote. So it took the product of the primary endosymbiosis, engulfed it, and that process is called secondary endosymbiosis. And that's just something I want to mention, is that the world we think about in terms of the biological world is really limited. You know, if you think of plants and animals and fungi, that's a teeny tiny part of biodiversity. It's really limited. And here's a diagram that kind of shows that. Look at this. Look at the bottom. Here you have animals and fungi, this group here. Here you have the first endosymbiotic event 1.5 billion years ago. It gave rise to, the red algae and the green algae and the land plants. And then you should also notice, you don't need to know any of these names, of course, just notice that some of these After the primary endosymbiotic event gave rise to these things, sometimes those, when they were single cells, they were then engulfed by something else and gave rise to all this other stuff. You know, chromera, apicomplexans, dinoflagellates, cryptophytes, excavata, all these things that you've probably rarely heard of, and certainly none of us would know what they were if we saw them. So our plant definition is the Viridiplantae. They split from the green algae about 1,500 million years ago, 1.5 billion years ago. There were both single-celled and multicellular forms. At that time, they were marine. And they lived, many of them, in moist environments near the ocean shores. And then, all of a sudden, 500 million years ago, there was the colonization of drier environments, that is, the terrestrial world. These organisms moved from a marine environment to the terrestrial world. And they had a number of adaptations, including a cuticle, which is a waxy covering to keep them from drying out. They developed vascular tissue. They developed a relationship with fungi, which we'll see later on. Almost all plants have fungi associated with them in their roots. They develop seeds and pollen. And since colonizing the land plants have evolved into about 290,000 species, those that exist today still. I don't know if that seems like a lot to you, but there are over 300,000 species of beetle. Of course, they're the big group, right? But, you know, a million species of insects, or who knows how many million. But 290,000 species of land plant. Not impressive, but not enormously impressive. One of the challenges that we have is understanding deep time. Our brains don't do very well with deep time. That is great big long time periods. And so here's a clock diagram of deep time on the Earth. And there are a couple of things I just want to point out here. The origin of the solar system is, and the Earth, is around 4.5 billion years ago. And then shortly thereafter, in a sense, 3.8 billion years, prokaryotes first appeared. Then photosynthesis was invented by prokaryotes, that is the cyanobacteria, for example. And somewhere around 2.5 billion years ago, the atmosphere then increased greatly in its amount of oxygen. So right now, oxygen forms about 21% of the atmosphere. In this early time, there was very little oxygen in the atmosphere, very little. And the oceans had a lot of iron in them. And once the atmospheric oxygen, really picking up here around 2.4 billion years ago, really picking up, once it dramatically increased in the atmosphere, the oceans then had a lot more oxygen in them, and iron decreased, and different sorts of organisms were able to live. Multicellular eukaryotes appear around 1.5 billion years ago. And then this amazing event, 500 million years ago. First of all, there was the invasion of the land by plants and animals and fungi. Second, there was, in marine and terrestrial environments, this explosion of types of organisms, and most famously of animals. Every phylum that you know, every phylum that has ever existed, appeared, except for a few earlier, appeared 500 million years ago in an event called the Cambrian explosion. This is one of these magical sorts of, in a sense, events where things happen really quickly in geological time. Sure, it was over tens of millions of years, but in geological time, it happened quickly, 500 million years ago. It got started, a little before that. So here's the oxygen revolution. And you can see around 2.4 billion years ago, the amount of oxygen went up dramatically, and this is because of the cyanobacteria. And then there was another little abrupt increase about 700 million years ago that might have been from true land plants. So I just showed you a lot of dates and things. What do I want you to know? Just the 4 here with arrows. So know that when the Earth formed, know that photosynthesis appeared 3.5 billion years ago. The first plastids around 1,500 million years ago, 1.5 billion. And then plants and red algae diverged around the same time. And then 500 million years ago, the colonization of land by plants and other things. So as you know from, if you took the 1010, Biology 1010, the evolution portion, you know that we define groups by shared characteristics. And these shared characteristics can be morphological, but now, today, we tend to use DNA information, sequence information, because it is so much more abundant and it's so much clearer in its storytelling. It's so much clearer in tracing history than morphology is. And this scheme here matches morphology, actually, and it matches DNA sequence data. So I'm going to describe to you what the morphological events are, what the morphological features, therefore, are at 1, 2, and 3. So everything that happened at 1 is shared by all the descendants here. So in this case, all plants. Everything that happened at 2 is shared by everything in the Streptophyta, the charophytes and the embryophytes. Everything that happened at 3 is shared by the embryophytes, which is a fancy word for land plants. Phylogenetic trees can be subtle things. You know, what's the reason for a curve here in this branch, for example? Is there meaning there? You know, does it mean anything how long that is? No. So I just want to show you that these two pictures are exactly the same. You know, wherever you put the 1s, 2s, and 3s doesn't matter. Those are exactly the same. All right. So first of all, number 1, what is the set of features that are shared by all plants? Number 1, starch as the main energy storage molecule. This is a big difference between plants and animals. As you know, animals use glycogen as the main energy storage molecule. Plants don't do that. Plants use starch. And starch is a polysaccharide. Poly means many. Saccharide means sugar. So starch is a bunch of sugars stuck together. And it is a carbohydrate, of course. And starch is a polysaccharide of glucose molecules, or glucose residues, they're more properly called, because it's not the whole molecule when you join it one to the next. And therefore, they come in these chains. So starch is made up of about 20% of simple chains, glucose, 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 and about 80% of branched chains. And the unbranched chains are called amylose, 20% of starch, and amylopectin makes up the rest, 80% that is, and they're branched. Why do plants do this? Well, you'd think they could just use sugar, wouldn't you? Why not use sugar? Plants make sugar from photosynthesis, so why not use sugar as the energy storage molecule? The problem with sugar is that it's highly soluble. So even though it's very reactive and it's easily metabolized, it holds water. So if you fill a cell with sugar, it's going to cause the osmotic potential in there to go up, or to go way down, that is. So the water will tend to go in. You add sugar to something, water's going to go in. So it causes all kinds of problems for the cell. If you turn that sugar into starch, then you've solved the problem, because starch doesn't have that same problem of being highly absorptive or changing the osmotic potential of the cell. So it's a fantastic solution. Starch is, as I'm sure you know, starch is easily broken down to glucose. This is something that anybody dealing with diabetes knows very well. Starch is very easily broken down to glucose. All right. The second feature of all plants is the presence of a chlorophyll, a pigment called chlorophyll B. All photosynthetic eukaryotes have chlorophyll A, and chlorophyll B just absorbs a different set of wavelengths. It's called an accessory pigment. And it takes energy from the sun and passes that energy along to chlorophyll A. But all plants have this extra chlorophyll that absorb slightly different wavelengths. The third feature is the presence of cellulose in the cell walls. Cellulose is another polysaccharide. So this is the second polysaccharide we've mentioned today. It's a major component of all plant cell walls, and it is unbranched glucose residues, different bonds compared to starch. Cellulose is the most common organic polymer on the planet. Cellulose is the most common organic polymer on Earth. Cotton is 90% cellulose. Cellulose is used to make paper. Cellulose is used to make rayon. You can buy socks made out of rayon. Other types of clothing used to make cellophane. The next feature of all plants has to do with the way that the membranes inside chloroplasts that hold chlorophyll are arrayed. And here's a chloroplast on the right, and this chloroplast has inside it these membranes that contain chlorophyll. And these membranes are called thylakoids. There's that weird word. That thylakoid is like a pancake, a stack of pancakes where each pancake is hollow. And the feature of all plants that's special is that these pancakes are in stacks. So these pancakes are in stacks, and this whole stack here is called a granum, so grana plural, and each thylakoid is, each so-called pancake would be a thylakoid. So the thylakoids are stacked into grana. So those are features shared by all plants. They unite the plants. The second set of features is the features that unite this other type of green alga, the charophytes, along with the land plants. And these charophytes are sister to land plants. They are not things that you would ever know. If you saw one on the street, you wouldn't know what it was. It was just a green thing. Here's chara on the left. Here's coleochaete on the right. We know from nuclear data, from chloroplast DNA data, and from anatomical structure that they are the closest relatives to land plants. The charophytes are, type of green alga. And just keep in mind that the charophytes we talk about, of course, are today living organisms. So they're not the ancestors of land plants, but they share a common ancestor with land plants. So what are the features that charophytes and land plants share? That is, the streptophytes. Well, first of all, it's the way the cells divide. So if you think of an animal cell dividing, it pinches in, right? And then the two cells separate. That's not what streptophytes do. That's not what land plants and charophytes do. Instead, they start in the middle and they pull themselves apart. So here is a cell plate, and that cell plate in the cell is then pulled apart by these microtubules. And then microtubules are called the phragmoplast. So that's how cell division occurs in the streptophytes. So cell plate and phragmoplast. The second is this really cool feature called plasmodesmata. And these are simply extensions from cell to cell to cell. And these extensions allow fluid to move. So let's draw this. All right, so we're going to draw 3 cells in a streptophyte. Here we go. Well, okay. Well, I was going to draw this with you. Goodness, that's never happened before. Okay, it didn't happen last class. If we drew this together, this is what we would draw. Fortunately, I took a picture of what we just drew. So here are 3 plant cells in a row. What I want to show you is that plant cells have cell walls. So the cell wall is out here. That's it. I got 2 lines to show the inside and outside of the cell wall. Lots of cellulose in there. Inside is the cell membrane. You all know cell membranes from the previous term. And this cell membrane here is inside the cell wall of these streptophytes. And then inside that, there's often this organelle called a vacuole, so I put a V there. And it's just for holding various kinds of stuff. And This red here, this third line in, is the membrane. You can see that the membrane goes from cell to cell to cell through these little pores. And so this ability of fluid to flow from one cell to the next is not found in other organisms. You wouldn't find it in animals. It's found in streptophytes, that is land plants and charophytes. And this opening here, this passageway from one cell to the next inside the membrane, is called a plasmodesma. The plural is plasmodesmata. So that is a feature of streptophytes. I'm sure there's a simple switch on that document camera that just needs to be engaged. The next feature is sperm structure. I'm just putting it here for completeness. I'm not going to talk about it at all. And then peroxisome enzymes. I know that you've learned about peroxisome enzymes in the cell biology part of this course, but plants have a particular, or streptophytes have a particular set of them that are special to that group. I'm not going to talk about that any further either. The next is that the cellulose that's made in the cell wall is made in all of these streptophytes by a rose-shaped complex, or rosette-shaped, same thing. So you know a rose window in a cathedral, that's the sort of shape here. And you can kind of see this here. There are two of them shown here. And this is in the cell membrane, the living part of the organism in the membrane that makes the cell wall to the outside. It's in a rose-shaped structure. The next feature of streptophytes is sporopollenin. And sporopollenin is one of the great inventions of life. Sporopollenin is a very durable polymer. Its chemical composition has not been fully worked out. It is the outside of plant spores. It's the outside of pollen. It's highly stable, chemically inert. It persists for a very long time in the environment. And it protects what's inside, pollen, for example, or spore, from desiccation. This is how you spell desiccation, by the way, one S and two C's. From decay, from attack by bacteria, from attack by viruses, from attack by molds, etc. Why would you move to land? Why, if you've got this happy, watery environment, a marine environment, why would you move to a terrestrial environment? The land plants are the plants that have taken over. They're by far the most species rich. They're by far the most, they cover the most land. What would be some advantages? There are some obvious ones. You could say, well, air filters less sunlight than water. So there's more light for photosynthesis. Air also has more CO2 than water, so more fuel for photosynthesis. Early terrestrial habitats wouldn't have the pathogens, the various problems that impinge on your fitness, predators, herbivores. And terrestrial soil is far richer in nutrients than aquatic soil is. So these are some benefits of land. This is a foreshadowing. I'm not going to go through this now, but you should come back to this slide later on in the course and say, how do I understand lignin, or how do I understand what a cuticle is, or vascular tissue, and that sort of thing? Each one of these is an adaptation to life on land. An adaptation is a feature that enhances fitness compared to a previous feature. So, for example, in water, water is highly supportive, as you know. Land is not supportive. So turgor pressure has evolved. Cell walls with lignin. Lignin is a polymer that makes wood woody. And things like xylem and things like stems. So come back to this later and have a look. It's just a foreshadowing. All right, finally, the land plants. This is what we're going to spend the rest of the plant part of my section on, the land plants. First of all, a waxy covering covering the whole organism. This prevents desiccation, prevents drying out, or helps slow it down. Second, something called gametangia. These are multicellular sex organs that have a set of sterile cells around them, and inside they make the gametes. So this set of sterile cells is why they're called jacketed. The important part is that they're multicellular and that they're called gametangia. These are the sex organs of land plants, and they come in two types. There is the antheridium. That's the male type. It makes sperm. One of the most important concepts that you're going to work on today and next time is that these sex organs are haploid. The sex organs that you think of that make sperm and egg in animals are not haploid. They're diploid. When your goldfish makes sperm, it makes sperm by meiosis. When your goldfish makes an egg, it makes an egg by meiosis, right? Land plants do not do that. Land plants make sperm by mitosis. That's because the sex organ is already haploid. The sex organ is already haploid. So this male sex organ, called an antheridium, is already a haploid structure. And this is mind-bending, I know, it's warping. Okay, here's the female sex organ. It's called the archegonium. Land plants have an archegonium. The archegonium makes an egg. The egg is made by mitosis because the archegonium itself is haploid. You can't undergo meiosis if you're haploid, right? Not possible. You have to be diploid or something higher, right? So the archegonium is the sex organ that makes the egg, and it's haploid. The next feature, so this leads to this feature called the embryophyte condition, and that's where land plants get their name. So embryophyte means embryo is embryo and phyte is plant. So what this means is that the mom, the thing that makes the egg, is going to keep Junior, and Junior is going to develop on mom. So imagine a world where mom makes an egg, the egg gets fertilized, and the egg then goes off somewhere and starts to divide and develop. Okay? That world exists. Lots of things do that. Land plants don't do that. In land plants, the egg is fertilized, and mom keeps it. So the archegonium, which is on another part of the plant, the archegonium, the sex organ, keeps it, and Junior starts to divide and divide and divide and develop. That's the embryophyte condition. The embryophyte condition is that the zygote or embryo is retained in the maternal tissue and divides. Here's a picture. So this is an archegonium, this maternal tissue, haploid. Fertilization has occurred. The embryo, which is diploid, starts to develop by cell division. So that's the embryophyte condition. The final feature is the coolest one of all. And this is the one that really, I think, is a challenge to get. But once you get it, you'll think this is really neat. And this is called alternation of generations. An alternation of generations is defined in the following way. First of all, what is the definition of sexual reproduction? In biology, sexual reproduction means the alternation of a haploid and a diploid state. So haploidy is caused by meiosis, and diploidy is caused by fertilization. Meiosis, fertilization, meiosis, fertilization, meiosis, fertilization. Causing haploid, causing diploid, causing haploid, causing diploid. That's sexual reproduction. That's what it is. In the case of your goldfish, what are these things? The diploid is your goldfish, right? Or your cat. What's the haploid? The sperm or the egg? How many cells is a sperm? 1. How many cells is an egg? 1. So land plants do something different. Land plants have both the diploid and haploid phases multicellular. they're weird. And so alternation of generations is defined as a multicellular diploid and a multicellular haploid phase. This multicellular diploid and multicellular haploid are the two so-called generations in alternation of generations. They have specific names. The diploid is called a sporophyte, and the haploid is called a gametophyte. The diploid is called a sporophyte. The haploid generation is called the gametophyte. All land plants do this. And this feature actually arose not only in land plants, it has arisen in other groups as well, independently. So there's something about this that's beneficial in various cases. These alternating generations are the gametophyte and the sporophyte. So first, the gametophyte. Break this word down. It often helps to look at the word and figure out its etymology. And this gametophyte, phyte means plant, gamete is gamete, so this is a plant that makes a gamete. So that's easy, right? A gametophyte makes gametes. So the gametophyte is what's going to make the sperm or the egg. The gametophyte is haploid. So you know it makes sperm and egg by what kind of cell division? What is it? No, it's haploid. It's making a haploid product. So it can't do it by meiosis. It has to do it by mitosis. Because if a haploid is going to make another haploid, it just has to divide. So that's mitosis. Yeah. So a gametophyte makes gametes by mitosis. So it's that generation. And a sporophyte makes spores by meiosis. So when you leave today, you need to walk down the street and sing to yourself, do it out loud. Land plants make spores by meiosis. That is the weirdest concept I'm going to mention today, maybe the whole term. Land plants make spores by meiosis. It's weird. They don't make eggs and sperm by meiosis. They're not like the goldfish. They make spores. And we're going to see next time how the spore then grows into the gametophyte and how this all works. So this idea, alternation of generations, is a core concept in land plants, and we're going to explore it much more fully next time. See you then.